The overall goal of the following experiment is to develop a novel DNA extraction technique that combines mechanical DNA homogenization with PCR inhibitor removal and the gold standard enzymatic extraction method to environmental samples along the poultry production continuum. This is accomplished by first dividing out an environmental sample into microcentrifuge tubes containing a mixture of beads to effectively homogenize the complex environmental samples. The second step is to place the tubes into a high-powered homogenizer to efficiently release nucleic acids from the bacterial cells and the environmental matrix. Next, the homogenate is used as the starting material for the gold standard enzymatic DNA extraction method through the PCR inhibition binding phase. The final step is to place the samples into a DNA extraction robotic workstation to provide the user with extracted DNA samples amenable to a host of downstream applications. Ultimately, quantitative PCR and microbiomic analyses using the Illumina-MiSeq platform were used to show that this novel hybrid method provides the best combination of qualitative and quantitative assessments of total bacterial communities compared to processing samples with the mechanical or enzymatic extraction method alone.
This method provides insight into the analysis of microbial communities along the poultry production continuum, but it can also be applied to other agricultural systems, such as swine, cattle, or crop production that contain similarly complex environmental samples. Demonstrating this procedure today will be Caitlin Griffin, a student in my laboratory.